Our next speaker is Jesper Andersen, who is joining us from the University of Copenhagen.
So first, let me thank the foundation for inviting me, and Ben and Nabil for also inviting me and organizing this great program. Okay, so it's quite humbling to actually be in a meeting like this, because as a scientist and a non-clinician, you never really get to talk to patients. And yesterday and today, I've been talking to quite a few of the patients that have been around here, and it's very moving stories that they can tell, which makes you think that we're not here just to publish the next paper, but actually to try to find cures altogether for these patients. So on top of that, I also have to confess some things. If you're confessing that I'm not really a mouse geneticist, not by far at all. I typically study human molecular pathogenesis and translational genomics, so I might have modified the title a little bit compared to what I was originally invited to do. So my title is Translational Genomics, Perspective of Cholangiocarcinoma from Mouse Models. And I will start by giving a background to the animal models used in cholangiocarcinoma, and a lot of them we have already heard from way more suitable speakers on this topic than me, such as Stuart Forbes and Holger Hillenbrand, and after me, Nabil will be giving a talk about it. So many of the models are probably seen in this as well, and I'm not by far going to say that it's a complete list of models, but there's a lot of models now finally for cholangiocarcinoma. After that, I will give a highlight of novel translational approaches with therapeutic opportunity, and we already heard about the not-canonical signaling, and I will also get into that, as well as targeting the receptor tyrosine kinase pathway, the ERB2 or the HER2 pathway. So first, some of the background. So I call these historical. That's maybe a little provocative. Animal models in the biliary tract, the models of biliary tract cancers. And you can basically group the first group here in the red bar as carcinogenic models. They were really the only thing that was available to study cholangiocarcinoma when I started looking at cholangiocarcinoma 10, 12 years ago. And Alphonsus Sirica's BDE new cell transplantation model in the rat was kind of the golden standard. That was very quickly followed by genetic models, of which we have already talked about some of them. And now also a number of other genetically engineered mouse models, of which I will go through pretty fast. The SMAD P10, P10 KRAS, KRAS P53, PIK3CA, YAP activation, and mutated AKT YAP pathways. And you kind of already see a pattern here that is P10, KRAS, P53, and YAP that are the overall line of all of these models. There are also a number of progenitor cell-derived promotion models of intrahepatic cholangiocarcinoma, of which the FIGROS was the first one. The P53 Nestin story published a few years ago, as well as the story from Nabil's lab. Maybe he will be talking about that in a moment, I don't know. Looking at the IDH and IDH2, we have definitely heard about this model earlier today. Then there was also the hydrodynamic injection model. We also heard about that, which in general is using the Sleepy Beauty transposinase and selectively adding in your oncogenic target, and by a very high volume injection in the tail vein of the mouse, basically infusing a cardiac arrest in the mouse, breaking up the mice again, you get tumors basically all over the liver by uptake into the hepatocytes. So not necessarily the best type of model for studying therapeutic responses, since you have tumors everywhere in the liver, and that's typically not how you see a patient. However, this is data from our lab in collaboration with Dr. Matter at the University of Basel. We looked into the KRAS family, both KRAS, the KRAS, the NRAS, as well as the HRAS, and used NOTCH, activated NOTCH as a control. It was already published, giving intrahepatic cholangiocarcinoma at 100%, whereas these models are not 100%. It's a mixed type, both hepatocellular carcinoma and cholangiocarcinoma, and HRAS mainly, or around 100%, actually gives hepatocellular carcinoma. And down here you can see the double stain with the K19 indicating the fact that this is intrahepatic cholangiocarcinoma. And I'll get back to this model a little later. We also, a few years ago, published the primary liver carcinomas can originate from different cell types, a little like what Holger Hillenbrand talked about earlier, of the dedifferentiation of hepatocytes to cholangiocytes. And in this model, we used the HRAS activation in the different steps of the hepatic differentiation, both hepatic progenitor cells, hepatoblast, and adult hepatocytes. And we saw a continuum of, to our surprise, we saw a continuum of primary liver cancers, kind of an undifferentiated epithelial-like derived from the hepatic progenitor cells to a more cholangiocyte-like type tumors from the hepatoblast to from the adult hepatocytes, a more hepatocellular carcinoma-like type tumor. And in total, the HRAS SV40 led to a strong induction of the c-Myc, and c-Myc has been found to be central in the hepatic or hepatocyte dedifferentiation. So the hydrodynamic injection model has been used quite extensively over the last few years. And here, I'm not even sure this is fully extensive anymore, but published in 2014, at that point, least extensive list of the various combinations of oncogenes or silencing of tumor suppressors, leading to liver tumors using this type model. But as can be seen, quite a few of these oncogenic models or models in the hydrodynamic injection model leads to intrahepatic cholangiocarcinoma. Actually, only activation of NOTCH and in combination with mutated AKT leads to intrahepatic cholangiocarcinoma, as well as two models leading to a mixed hepatocellular carcinoma and intrahepatic cholangiocarcinoma. The rest leads to hepatocellular carcinoma. So that leads me to the role of NOTCH activation in intrahepatic cholangiocarcinoma. So NOTCH, as I've already been discussed, the NOTCH receptor expression as well as regulation is important for normal bile development, but we have seen already from Dr. Hillenbrand that cholangiocarcinoma overactivation of NOTCH together with AKT leads to intrahepatic cholangiocarcinoma. That was also followed up with another lineage tracing experiment from the center lab a year later, I think it was. So, but it's not involved in human cholangiocarcinoma. That brings me to the second part of the talk, which is to highlight the novel translational approach, or at least the translational approach of therapeutic opportunity, both looking at the NOTCH canonical signaling as well as the receptor tyrosine kinases. But first, the NOTCH canonical signaling pathway. So, based on the literature and the publications around 2013 of activated NOTCH and AKT leading to intrahepatic cholangiocarcinoma through a dedifferentiation of hepatocytes to cholangiocytes, we thought that, okay, let's look at our database of cholangiocarcinoma gene expression data from the patients. And we simply picked out the 4 different receptors and grouped them. To our very big surprise, we were able to group them in 4 very distinct groups, with each of these groups having a high expression of either of the NOTCH receptors, but never an overlap between the expression pattern of either NOTCH1 and NOTCH2. So in the case of NOTCH1, high expressing patients, they have the absolutely poorest overall survival looking at 5 years compared to NOTCH2, 3, and 4. And that was an even stronger poor survival, if you will, than how our original poor and good survival classification of these patients were devised when we did unsupervised clustering of the patients. That made us pull out the majority of the NOTCH signaling pathway here the NOTCH receptors, the NOTCH ligands, the NOTCH targets, or at least some of the targets, and the gamma secretase complex. This scheme was then devised for overexpression in the tumors compared to matched surrounding. So the more to this direction, you have higher tumor expression. The more towards the ceiling of this, you are more significant. And as you can see, a very big proportion of the entire network is overrepresented in the tumor significantly as well. And it can probably also be appreciated that the brown or brownish colors, the gamma secretase complex, is really among the more upregulated and more significantly upregulated members, as well as the one in the circle is the active component of the gamma secretase complex in castration. So the canonical NOTCH signaling pathway is, I would say, definitely deregulated in intrahepatic cholangiocarcinoma. That led to the question, and we have already heard that in Stuart Thorpe's talk just minutes ago, if gamma secretase inhibitors would be effective in attenuating the NOTCH activation. And true, gamma secretase inhibitors has a lot of side effects to be considered. However, if one could first see the proof of the concept, we could probably look after another inhibitor downstream. So before doing any experiments with the gamma secretase inhibitors, we wanted to make sure that that NOTCH it truly was activated in these patients. So we did both stains of the total NOTCH one as well as the activated part of NOTCH, the NICD component that we have already heard about. And based on our initial nearly historical classification of intrahepatic cholangiocarcinoma patients in poor and good prognosis, we can see a clear overexpression of the intracellular domain of NOTCH in the poor prognosis group, and we did this both by immunohistochemistry as well as with hematoxylin staining, seeing that the activated NOTCH really do get into the nucleus. And that was, of course, significant between the two groups of patients. That led us to, as I said, acquire the gamma secretase inhibitors, and in this scheme, 13 different intrahepatic, or 13 different cholangiocarcinoma cell lines. We acquired 2 gamma secretase inhibitors with the lowest needed concentration for IC50. We treated over 3 days and 7 days, various different concentrations, and we're able to group the cell lines based on overall drug sensitivity, from the most sensitive, meaning the fact that they inhibited the activation of N1, that's not one of the intracellular domain in this for simplicity, to cell lines like the KMBC and the SNU478 that mainly were very close to being resistant to the treatment. So is canonical NOTCH signaling a possible therapeutic target, though still thinking of the various side effects that it may have? So we made a mouse out of this, basically just xenotransplantation. As I said, I'm not a mouse geneticist. And we chose gamma secretase non-responder or resistant cell line from the previous screen, as well as a gamma secretase responder cell line, and transplanted the cells, both control, meaning not treated in one flank and treated in the other flank, and the same thing for the responder. Control is not treated, and treated is on the opposite flank. So matched in the same mouse, and then multiple mice, of course. And as you can see, or probably appreciate, the gamma secretase responder cell lines from the previous screen truly also in vivo showed response with no further growth compared to the control-treated, transplanted cells. From these tumors, from these mice, we extracted the tumors, and we actually did it both for the non-responders and responders, but for simplicity, I'm only showing the DSI responder signature, which then recapitulated the NOTCH signaling pathway using gene set enrichment, and looking at the core networks in the gene set enrichment, we see all the things that we would want to see, basically, if the treatment was successful and precise for inhibiting NOTCH activation through gamma secretase. We see NOM that's downstream from it, the encastrin itself, the activating component, as well as several different other components of the pathway. That leads me to the second part, or the third part of the talk, targeting the receptor tyrosine kinases. That was actually devised quite a long time ago, as was mentioned earlier as well, all the way back to 2012-13, when we originally did the patient classification, followed by Daniel Assier's classification. And then we combined the two classifications, and overall we see a pretty good correlation between the two studies. And overall we see an overexpression of the ERB family, as well as the KRAS mutation being in the proliferation group, or the poor prognosis group of patients. We also confirmed that by looking at a type of a multiple Western blot protein array type study, and again in the poor prognosis groups, in two different patient groups here, good and poor prognosis for either of the groups. We see an overexpression of many of these components in the pathway, as well as both the ERB pathway, COX-2, which will become important in the talk, AKT, as well as STAT and IL-6, as we have heard about earlier in this meeting as well. We confirmed this by immunohistochemistry of the patients, where we, we did immunohistochemistry of the patient still based on the good and poor prognosis from our original paper in 2012. CO expression of HER2, MET, EGFR, with CO expression of COX-2, specifically for both specifically for the epithelial part of the tumors. So do we actually see an oncogenic addiction? It's a very dangerous word to use in this context, but anyway, do we see an oncogenic addiction to tyrosine kinase pathways, and will one be able to treat that? For that reason, we devised this, if you will, preclinical study design, where we used a KRAS mutated cell line, HUCCT1. All the cell lines we have in the repository are fully worked through for genomics and epigenomics, as well as mutations, so we know exactly how they match to patients and can integrate that information as well. We randomized after we could palpate the tumors, and we randomized into 4 different groups: control, trastuzumab, celecoxib, and celecoxib trastuzumab. And as I mentioned, that's where celecoxib comes in, or the COX-2 overexpression in the poor prognosis group. Then we have a treatment phase that ran for 4 weeks, and that is described here. We did a withdrawal to know if this was cytotoxic or cytostatic effect, and then we get into mouse surrogate survival until the veterinarians say you have to stop the experiment, not to be cruel with the size of the tumor and so forth. So in this slide, I will summarize basically all the data from that animal study, where we see an increased effect, where we asked if we saw an increased effect of the targeting of HER2 in combination with COX-2, HER2 in combination with COX-2 inhibition. And and here you can see the control, celecoxib, trastuzumab, celecoxib combination, and totally the combination are really very homogeneous group compared to the other single arm, as well as the control that has much more variability in the tumor size. We analyzed the inhibition rate over the 4 weeks of treatment, and for only the celecoxib trastuzumab group do we see an increasing trend of the treatment, whereas for the 2 single drugs, celecoxib or trastuzumab in black, do we either see a flat or downtrending effect of the initial effect seen by treatment. That led to then the withdrawal phase, the 5th week of the study, where we do see a small loss of the inhibition of the drug, but not a complete crash compared to the 2 single treatment. So by far, I'm not saying that we can actually treat the mice for the tumors, or it's a cytotoxic effect, but there is a mix between cytotoxic and cytostatic effect in this model. So that translates into survival post-treatment of HR or hazard ratio of 6.3, which is pretty good statistical difference between the control and the double arm. We excised the tumors also in this part of the study, and were able to generate a tumor response network, which recapitulated what one would expect to see when treating with these drugs, MAPK and mTOR pathway. Particularly the mTOR pathway then led to our next part of the story, and that was asking what about AKT in this setting? That led us to use the same model, using a preclinical application of AKT or the Merck's drug MK2206 in intrahepatic cholangiocarcinoma. We first again wanted to validate that activated AKT or the serine 473 truly is upregulated in the poor prognosis or increased in cholangiocarcinoma. And then in total, we saw in the same model as before an inhibition rate of 81% between the control and the treated arm, and that also translated, of course, to a different size in the tumors. And to steal a little bit of nomenclature from the clinicians, we actually saw a complete response in 3 cases where we basically could not find the tumors anymore. We saw kind of a partial response, if you will, in 3, and steady disease in 2, where the tumors never moved at all from the time when we started the treatment. And this is how we devised the dose as well as how we gave the treatment. Again, we excised the tumors, and we performed we performed a gene expression arrays on these tumors compared to control. We were able to devise a response signature, and that response signature was enriched in 2 other studies, but in hepatocellular carcinoma, since there is no study in intrahepatic cholangiocarcinoma in that regard. Both the study from Hoshida of AKT activation signature, where there was an enrichment of the genes we identified in this after this treatment, as well as from Sukman Rossi's group in France. There is I actually don't know if how the trial is going, but there has been at least an ongoing phase 2 trial from Merck in clinicaltrials.gov, and that would be quite interesting to see what the data is. We also did a lot of sequencing in intrahepatic cholangiocarcinoma patients, and this is just a small take on that. Is the screening of the mutational frequency of cancer-related or 48 cancer-related genes in 155 fresh frozen intrahepatic cases. We have a cutoff at 500 copies of the variant read, and there has to be more than 5% allelic variant call for calling it. And that brings this slide because I really just want to emphasize the fact that the pathways that I've been talking about are just a few of these, and what has been actually talked about at the meeting is very few of the ones that are really here. There are a lot more going on than what is currently being studied. So I talked about the ERBI, the HER2, AKT, NOT, and KRAS was the model that we used or the patient model or the surrogate for the patients we used in the mice, if you will, for this modeling. So we have seen this before from Nakamura in 2015, Nature Genetics, where he walked through the basically upper GI in the ICDC study of more than 200 tumors. So it'll be interesting to see what happens when we get models for the FGFR2 fusions, if they are truly drivers or not. Definitely currently quite easy to treat compared to many of the other things we see in these patients. Another slide from another figure from his paper is considerably more complicated than the one on this side. Is an overall diagram of the different changes that are seen or networks that are seen based on mutations in all the tumors that they sequenced. But if one looks at it in a little more from above fashion, you really have receptor tyrosine kinases. You have DNA damage response type network going on here, as well as epigenetics. So you can kind of tease it down to 3 different things that are ongoing and that we will have to try to control. So this is the current genetic perturbations that are based on the anatomical tumor site in biliary tract cancers. So that's a lot of different models from the other people that have talked before me, and different translational genomics ways of treating in the mice and so forth. But a lot of these models we really lack to actually bring back to the patients and model if the tumors that are seen in the mice are really reflective of any of the patient groups. So a way to really just brush that aside completely is to use patient-derived therapeutic modeling or drug repositioning by having the primary tumor excised, generating either PDX or primary tumor cells, as we have done here, and then over time make sure that there is no fluctuation of the genetic profile of these cells that have been excised. So in comparison to the patient's own surrounding tissue for germline, matches to the tumor, and then follow them on passages as you're generating the primary cells. So that can be used either in the mouse for therapeutics, or we can use it for drug sensitivity and resistant testing using already approved FDA drugs to see if we can, as the name says, drug repositioning, if we can identify drugs that can be used in intrahepatic cholangiocarcinoma or any other type of cholangiocarcinoma for that matter. So can you teach an old dog new tricks, basically? So can you simply just steal from other therapeutic areas and then positioning that into cholangiocarcinoma? And we're currently doing a lot of that screening in both primary as well as established cell lines to see if there are specific patterns in that type studies. So thank you very much for your attention and for the invitation again. This is my lab that started in 2014 after a number of years in US. This is a lot of great collaborators. Some are here, others are back in Europe, and all the funding agencies. And then one last remark I want to say is that though very little is actually happening in cholangiocarcinoma in Europe, meaning me and 9 people from 9 other countries, last year established a small group of people with an interest in cholangiocarcinoma called the European Network Study for Cholangiocarcinoma. And this year in Spain, we're going to have our first meeting in May. So Stuart Forbes here is also actually a member of that and an original founder of the meeting. Thank you very much.
Questions? And then again, back to Notch, when you were describing the clustering of patients according to Notch receptor expression, you had the 4 categories corresponding to the 4 receptors, but it looked like everybody was overexpressing one receptor. Did I read that correctly, or was that a function of how you
No, you
presented the
actually
data?
read that correctly, and that is really very surprising to us, and I think we need more data actually. So I'm still collecting. If anybody has first frozen tumors, I would be happy to receive them, and particularly distal cholangiocarcinoma is something that I'm liking a lot to map from intrahepatic and all the way down to the gallbladder what really goes on in the gene expression space. So today we are past 200 fresh frozen tumors that have been profiled. What I showed you was the original number of 104 patients from the gastroenterology paper in 2012. So it's going to be interesting to see if we can redo that clustering. It's supervised because we just picked out 4 receptors, and then they all fall into to one or the other category.
Thanks, Jesper. A couple of lifetimes ago, we were looking at the inflammation Notch story with INOS. So INOS is richly expressed in PSC, for example, and it induces Notch. So have you looked at INOS Notch correlations in any of these tumors?
Actually, no, I have not looked at INOS, but of the 104 patients, I think there are 3 that are PSCs, so there's really not very many PSCs. But still, thank you. I should look at INOS.
I enjoyed the talk. I was just wondering, the in vivo AKT inhibitor data was actually quite a dramatic response. I'm just wondering what model was that? That looked like a real bona fide sensitivity. What
model?
Yes.
It was the same as for and that I used for the ERB part. It was HU-CCT1 KRAS mutated cell line that was transplanted, xenotransplantation. So not no fancy mouse genetics.
Yeah, got it.
I have no understanding of that.
Yes,
So okay, thank you.
Thank you.